Okay. Hi. Good afternoon, everybody. And we are going to move on from our discussion about cell division, and especially about meiosis, the type of cell division which gives rise to gametes. And we're going to start talking very briefly, unfortunately. It has to be brief, but about the science of genetics. And that is to examine the principles of inheritance. How do we inherit certain characters, certain traits from parents, how they pass to offspring, and how do they move from the offspring to their own offspring? Well, this is a question which has troubled human beings for a very, very long time. And we only had a very poor understanding of the principles of inheritance because there are some strange things happen. The results are not always immediate. They don't appear to be immediately predictable. They become predictable when we understand some clear principles behind the mechanisms of inheritance. And they become even clearer when we link those principles to what we know, especially about meiosis. Because if you understand meiosis and what happens during meiosis, you very easily can explain the simple principles of inheritance. So the first person to actually formulate this understanding was Gregor Mendel, who was an Austrian monk. He was a very keen naturalist and a very avid gardener. That was actually his job at the monastery where he lived. And he was a beekeeper. His initial interest in inheritance and the principles of inheritance was actually with the idea that he wanted to improve his beehives. Unfortunately, bees are a very bad organism to work on if you're interested in inheritance, because all members of a beehive are so incredibly closely related. They're basically all daughters of the queen, and they basically all clones of one another, and they don't reproduce themselves. Only the queen and some of the male bees, the drones, will reproduce. Very, very difficult thing to work on. Mendel had observed, of course, as human beings have for a very, very long time, that farmers could breed organisms, both animals and plants, to amplify certain characters, certain beneficial characters. So farmers had, over time, had bred horses which were huge, plow horses with huge muscles, very heavy, rather slow, but very powerful. And they also bred horses which were fast, easy riding, etc. And there was certainly a certain amount of predictability in taking parents with certain characters, breeding them, and having offspring with similar characters. It was also obvious that farmers, especially, could select characters and amplify them in a population. So Gregor Mendel looked at all of this and tried to start formulating some really scientific principles that would make breeding predictable. And just one sec. It's very important to realize that Mendel knew nothing about chromosomes. He knew nothing about DNA. He knew nothing about meiosis or cell division or any of that stuff. All of his work was based on observation, and very careful observation. It is obvious when you read his journals and also when you look at his experiments that he did a tremendous amount of preparatory work before he ran his experiments. So he set very clear parameters around what he studied. I'm emphasizing that because the initial principles of genetics that we learn through Mendel are perfectly valid. They certainly apply. But genetics is actually a lot more complex, it turns out, than Mendel's simple ideas. But nonetheless, we start with Mendel's ideas, And we would then amplify those in the rest of the study of genetics. Mendel's ideas give us a true, valid framework to study and understand the principles, at least, of genetic inheritance. So I'm going to show you this diagram very quickly, just to link the sorts of things that Mendel saw with what we know about meiosis, for example. So we're going to find out that Mendel studied pea plants, and he in particular studied characters of pea plants which occurred in pairs. So the pea plants were either purple or they were white, the flowers. There was no in between, there was a clear-cut character that he chose, purple or white, and he grew those plants. He could breed, he could let them get fertilized and then produce offspring, and then look at the offspring and see what their characters were. So here's what we're going to observe. Let's just have a look. Here's a purple flower, and we know that this purple flower has a whole lot of chromosomes, but the chromosomes occur in pairs, and the character, this flower color, the gene for that resides on a chromosome, and they're homologous chromosome with the same gene at the same place. The places where they occupy there is called the locus, and the character is flower color, and in this case it is purple. We say that this is the purple allele because there are other color alleles at the same locus that can occur at the same locus. So this is, Mendel chose this. This is a plant which has two identical chromosomes, an identical maternal and an identical paternal. What are the possible gametes that could arise from this? Knowing about meiosis. Well, in meiosis, we take this cell, and if it's going to produce gametes, it's going to segregate them, right? We're going to end up with 4 cells with half the number of chromosomes. There's a homologous pair. Of the homologous pair, each of these cells will have one chromosome. In other words, each of them will carry one of these alleles here. Let's artificially fertilize our flowers. We're going to take pollen, sperm, from this purple flower. Okay. We're going to go to the white flower now. What do we have in the white flower? We have paternal and maternal chromosomes, homologous. Again, these are identical in this particular. I think Mendel chose that plant so that this was the case. He didn't know why, didn't understand about chromosomes, but that is why this plant predictably was white. Here's the allele for white. Here's the allele for white at the same locus here on these chromosomes. Let's fertilize this flower with the sperm. So the eggs produced by this plant, again, would be the same. We would segregate them, 1, 2, 3, 4, each of them with half the number of chromosomes. Now we allow fertilization to take place. We can actually do that fertilization ourselves if we wanted to, and that is what Mendel did. And now, when we fertilize, look what we end up with. We'll end up with one of that allele, one of that allele. Diploid again. Diploid. But now remember that this can go on to reproduce. And now look what happens when we segregate these alleles in the next generation. We end up with a mixture. We end up with 2 like that, and we end up with 2 like that. This may not make a lot of sense now, but when we're going through the examples that follow this, come back to this diagram. Come back to this diagram and remember meiosis. And if you can do that, if you continually refer back to this diagram and remember the processes of meiosis, everything will become clear. Everything that we're going to talk about in this section on Mendelian genetics is basically explained here, believe it or not. Okay, so first of all, I've already told you, Mendel worked on garden peas, and he used to go to the farmers markets actually, and he used to buy pea seeds, and with various characters, and he would breed them very carefully. And so any heritable feature, like flower color, we'll see some others in a while, that we call a character. Okay, flower color is a character, but the variants of the character are called the traits. So purple flower is a trait. White flower, that's a trait. There are many, many of different characters like this that Mendel very carefully chose. He always chose something where there was a clear alternative, either purple or white, either tall or short, etc., etc. He very carefully chose these characters so that they contrasted. This is an example of the peas that he used. The advantage of doing this with peas rather than with something else like bees or cows or whatever is peas have a very short generation time. In a year, he could do several generations of peas, and he could do enormous numbers of offspring. He actually had assistants, so they could work with a large number of peas. And this is one of the huge advances that Mendel made, was he gathered data. He did it by the numbers. He counted very, very carefully what the results of all his experiments were. He didn't just look and hazard a guess. He actually put numbers down on paper and worked them out mathematically. The other really huge advantage that peas offered him was there is a choice of how you allow them to mate. Most horticultural bee and pea varieties that we use, in fact, if you leave them alone, they don't get fertilized by insects. They don't get pollinated by insects. They actually self-pollinate. So the plant actually fertilizes its own eggs with its own pollen. But you can artificially fertilize. You can pick pollen from one plant, take it, and fertilize another plant. So he had that choice. If you just left them alone, they self-fertilized. But if he wanted to, he could determine what fertilized what. So here's what he actually did. So here's also some very important terminology that we need to get clear. First of all, if he wanted to artificially pollinate, then what he would do was he would open up the flower here, and he would take pollen. He would take stamens, which are the male part, or the pollen, and he would cut them all off, and then to ensure that this plant did not fertilize itself, and he could then take pollen from another plant, and he would fertilize the female parts of the plant again. He emasculates the flower, right? He takes away all its ability to self-pollinate, and instead he pollinates it with the desired pollen. And he then goes and he has a look at the offspring. This initial cross here, this is called the P generation. That is the parental generation. And the offspring here is the first filial generation. and that is the F1 generation. If we then proceed to allow these to fertilize, or we fertilize them, then, and the next generation, that's the F2 generation is what results. Okay, so he's seeing a plant with a certain character. He's going to fertilize from plants with the corresponding character, and he's going to see what the results are. Well, very important. Again, I emphasize these two contrasting. The reason I hammer it is because we're going to come on later to hear some complications. The genes don't always occur in these pairs like this, but he just chose all the characters so that they would. He also was very careful that he used varieties which were true breeding. That meant if they self-fertilized, or if you artificially fertilized between plants with the same trait, they produced plants with that trait continuously. And we say they are true breeding. And if you want to realize why, it's because of the maternal and paternal chromosomes are identical for that character. So he starts with plants he knows are true breeding. So, and he would cross-fertilize them. Now, the prevailing theory at the time about inheritance was that inheritance was a process of blending. So if you took a parent with one character and you took the other parent with the other character, the offspring would somehow be a mixture of those two characters. If you had a mother who was short and a father who was very tall, their offspring would be of middling height. They'd be somewhere in the middle. Blending also said to Mendel that if he took a purple flower and he took a white flower and he crossed them, the results would be various shades of lavender, of diluted purple. So this is what Mendel was interested to know. He'd probably seen that this was not true, and now he wants to prove it. Okay, so he takes purple flowers, he takes white flowers. These are contrasting traits of the same character, and he breeds them. And he looks at the first filial generation, the F1 hybrids. And he doesn't get a mixture of lavenders. He gets purple, indistinguishable from the parental generation. Indistinguishable. They're fully purple. The purple's not diluted at all. There's no white at all in the first generation. So, where did the white go? We're not sure. So he took all of these, and now he allows them to self-pollinate, or he makes sure, if he does some pollination of his own, he makes sure that he cross-pollinates in all members of this generation. Easiest, just leave them to self-pollinate and then see what happens. And in the F2 generation, to his shock, surprise, there were two kinds of flower, purple flowers here and white flowers. The white flowers were relatively rare compared to all the rest. In fact, when he did all the counting, he ended up with, his initial experiment ended up with 705 purple flowered plants to 224 white flowered plants. That is approximately 3 to 1. The more you do this experiment, the more offspring you can get, the closer you get to a 3 to 1 ratio. That 3 to 1 ratio becomes extremely important because it allows him to construct a pattern of inheritance, and for reasons that you'll see in a minute. So he reasoned that the way in which this particular character worked was that you had one trait which was effectively a working trait. It produced purple. And the other trait, which is white, was simply an absence of any color. It wasn't that a white color was produced. It was that no color could be produced. And so if you had that purple allele present, the flower would be purple. But in the complete absence of that purple allele, the flower would be white. He called the purple a dominant trait, and he called the white color a recessive trait. So this recessive trait is not diluted by blending. It's still there. It's just that if there is present in the cell an allele for purple, then it dominates the allele for the white allele, and it kind of hides it. But when we come to when we allow these to self-pollinate, the white allele reappears because some of the plants now have no purple allele. There are a few which just have white alleles, and they are the white ones. These ones here have a purple allele, even if they also have a white allele. So this completely threw out the system of blending, the theory of blending now. With that simple experiment, Mendel has destroyed the theory of blending. So he then proceeded to look at a whole lot of other characters in plants. And he called these, he observed that these characters, these specific characters, could be passed from one generation to another. He called them heritable factors. We now know that those are the genes. There is a gene for purple. There is a gene for white. He called them heritable factors because he knew nothing about the chromosomes. He didn't know anything about the function of chromosomes. So here are all the characters which Mendel looked at, and all of them share this characteristic that they're either on or they're off. So there's a clear dominant and a clear recessive for each of these characters. One we've seen already, purple and white flowers. He looked at seed color. Seeds could be yellow, that was the dominant, or green, that's the recessive. You could look at seed shape. Either they're smooth or they're wrinkled. This is actually quite a complex thing, it turns out. This has to do with the amount of starch that is laid down in the seed, and it affects its osmotic balance. And the wrinkled seeds lose a lot of water and shrivel up like this, where the round seeds do not. He looked at pod color. Pods were either green or yellow. Pod shape, they were either like blown up or they were shrunken against the seeds, which he called constricted. He looked at the flower position. The flowers emerged from the stem on the sides, or they came at the top of the stem, which he called terminal. And he also looked at tall and dwarf plants. So each of these characters is either or, either or, either or. And if you breed them, if you cross-pollinate them, then the F1 generation will all show these dominant characters. You don't see any of the recessive. So the F1 generation, these are clearly dominant, all of these characters. F2 generation, if you breed them out, you allow them to self-pollinate or whatever, you end up 3-to-1, 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 3-to-1. When you look at these ratios, you may see that they're not quite 3-to-1. This is a biological system. You're going to have a little bit of variability. Maybe a plant got eaten. Maybe another plant got a viral infection and didn't appear or whatever. You're going to have a little bit of variability that you have to work with immediately you start working with biological systems. But the more you repeat these experiments, the closer and closer you get to a 3-to-1 ratio in the F2 generation for each of these contrasting characters. Why? Why is this? Why 3-to-1? What is about this 3-to-1 ratio? He came up with 4 concepts, which we can now understand very, very easily by thinking about what we know about the chromosomes and how they behave during meiosis. Okay, first, there are alternate versions of genes that account for the different traits. Okay, that is immediate for us. It seems obvious, but for Mendel, this was a revolutionary idea. There are alternative versions of something that is heritable that accounts for particular characters and particular traits of that character. So the gene for flower color in the peas exists in 2 versions, one for purple and the other for white. And we call those 2 versions the alleles. We say there are 2 alleles. There's a purple allele and a white allele. The important thing about the alleles, they reside at exactly the same place on homologous chromosomes. So they occupy exactly the same position on the homologous chromosomes, specific locus on a specific chromosome. So here, in fact, is what we understand. We now can go to the DNA of the chromosomes and we can look at it. And this is just to illustrate to you how this system of dominance here with flower color is working. Here is the chromosome, here the locus for the purple flower. In this case, they've put it on the male chromosome. And here's its sequence here, CTA, AAT, etc. And this produces, this gene is responsible for producing an enzyme in a metabolic pathway that ends up synthesizing purple, synthesizes the purple pigment. So this is a working gene, and it produces a purple flower. This allele here is for white flower, and sequence reads ATA, not CTA, but ATA, AAT, etc., etc. And it is non-functional. It doesn't produce the enzyme. So this metabolic pathway to produce the purple enzyme just doesn't work. So a flower, this gene on its own would produce a white flower. This one here would produce a purple flower. If we have one copy of purple and one copy of the white allele together in the plant will be purple, because one working copy of the gene is sufficient to color the flower purple. If, on the other hand, we had 2, if both genes were identical, it would still be purple. It's not going to be more purple. It's just purple. What is our third possibility? Our third possibility is that we have 2 non-functional genes. If both of the alleles in a plant are non-functional, the plant will be white. So we have a possibility: dominant plus a recessive, it's purple. 2 identical purple alleles, purple. 2 non-functional white alleles, the plant is white. This was an idea without knowing anything about this. This is already an idea which Mendel has clearly in his head, that there is an interaction between these alleles, between these heritable characters, which produces what we observe. The second observation, for each character, every offspring inherits something from the mother and something from the father. They inherit a heritable trait from mother, heritable trait from the father. They inherit 2 alleles. Well, we know that because we saw in meiosis how we produced gametes with half the number of chromosomes. So there's just 1 copy in a gamete, 1 copy in maternal, 1 copy in paternal. Put them together in fertilization, you end up with the diploid zygote, and the diploid zygote has 1 maternal and 1 paternal. And the homologous chromosomes have the same character at the same locus, even though they may be different alleles. So those alleles that they inherit here, they can be identical, and they can differ from one another. And we'll see the impact of that. Oh, sorry, let me see. So, and the third thing he observed, he observed this for his experiment, but in fact, he also kind of chose it so that this would be so. That the alleles he was looking at, all of the alleles he was looking at, existed in this relationship, that there was a dominant and a recessive, a clear dominant and a clear recessive, which could be identified. The dominant was, if it was present at all in the organism, that is the allele that appeared. And so the recessive could be silenced, but it did not disappear. The recessive, especially in the F1 generation, the recessive could be silenced, as we saw with the white flowers. But that recessive is not lost. It is simply silenced, and it can reappear in another generation. So the fourth law that he observed is the law of segregation. And that is the 2 alleles. Remember, we've got, in every organism, we have 2 alleles on the homologous chromosomes. They will separate during reproduction, so that only one is present in the gametes. So in egg and in sperm, there is only 1 of the chromosomes, either the maternal or the paternal chromosome. That is the law of segregation. So egg and sperm only get 1 of the 2 alleles that are present in the whole zygote, in the whole organism. And this we know because we saw in meiosis how the homologous chromosomes in meiosis get segregated to different gametes, to different cells. We ended up with 4 cells, each with 1 from the homologous chromosomes. That is how we explain this law of segregation. We can see how it works if we go back to meiosis. Okay, so here's a simple way to follow what is happening. And we need to understand, how is it that in this F2 generation, we end up with this 3-to-1 appearance of the dominant trait and the recessive trait. So in the F2 generation, that we need to understand what kind of gametes can the organisms actually produce. And here's a very simple way to do it. And it's called a Punnett square. And let's follow through. Here's Mendel's experiment. He takes purple flowers and he takes white flowers. Now, he knows that there is a maternal and a paternal copy. We know it's paternal and maternal. He just knew there were two alleles present. And in the plants that he chose, which were true breeding, they had 2 copies of the dominant heritable trait. In the white flowers, they had 2 copies of the recessive trait. And what possible gametes can they produce? Well, this one can only produce, I'm going to start talking about this as big P and little p. Okay. The purple is big P, big P. When it produces gametes, it can only produce big P gametes. That's the only possible. Just imagine this going through meiosis and segregating out into four cells. Let's say that this is male, 4 pollen cells. All they could have is big P. In the white flowers, remember, this is true breeding. If I allow this to self-pollinate, it's going to produce white flowers. It tells me there's absolutely no big P present at all. It's all little p. Its gametes, all it can have is little p. Now we're going to put them together. We're going to allow them to fertilize one another. What is the only possible combination we can get in this generation, in the F1 generation? We can only have big P, little p zygotes. Can only have big P, little p zygote. And there it is there. Big P, little p, but all of them are purple. Why? They're all purple because there is a gene present. The dominant gene is present. The recessive gene is still there. The recessive allele is still present, but it has no impact on what we observe. What we observe is called the phenotype. And in the F1 generation, the phenotype is always the same as the dominant. Okay. There's another thing. We have some terms that you need to learn. This here is called homozygous. That is homozygous dominant. This is homozygous recessive. Homozygous dominant, homozygous recessive. And this here is heterozygous. So it's heterozygote. It's a big P, little p. It's a mixture of the two alleles, big P, little p. So when we look at this here, we're going to ask what kind of gametes is it going to produce? We'll segregate them out, take it through meiosis, and end up with your 4 cells at the end, with your chromosome, with the independent assortment, etc., 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 and we sort them out. And all that we're going to be able to produce for gametes is big P, little p, little p. That's it. Big P, little p, little p. And half of the gametes will be big P, half of the gametes will be little p. Remember, think back to meiosis always. Just think back to meiosis and you'll realize why this is, why half of them are big P and half of them are little p. Okay, so now we're going to allow these to get together to fertilize and to produce a zygote, a new set of zygotes, which we're going to go out and we're going to count flower color. What is our predictable result? Well, here's what is called, this is the Punnett square. So this is a square, and it allows you to map out all the possible zygotes. So what are our gametes? Well, let's say from the female, we can only have big P and little p, right? Half a big P, half a little p of the gametes. The male, the same. The pollen, half of it's going to be big P, half of it's going to be little p. Those are our only 2 possibilities. This is what we do. We're going to work out all of the possible genotypes. This is the genotype. The genotype is the actual allele possession that a zygote has, right? So this zygote here, we've got big P pollen, big P egg. So we've got a homozygous dominant there. When we look at the other 2, however, here we've got eggs, big P, but here these sperm are little p. This is heterozygous, big P, little p. This one too, big P, this big P sperm, little p egg. big P, little p, heterozygous. So these are the genotypes: homozygous, heterozygous, heterozygous. What about this one? This one, we've got little p egg and little p pollen. And that is going to produce little p, a homozygous recessive. So what do we end up with? Big P, homozygous, little p, homozygous, and 2 heterozygotes. But when we look at the phenotype, when we look at what these genotypes actually produce, this heterozygote will be purple. This heterozygote will be purple. This homozygote will be purple. And the only one which is not purple is this one here, which is the homozygous recessive. Little p, little p. Count them up. 1, 2, 3 to 1. The phenotype, what we observe, the purple, is 3 to 1 in presentation, because you've got 1 homozygous, 2 heterozygotes, which are all going to be purple, and 1 homozygous recessive, which is going to be white. The same thing applies if we do any of these. If we take any of these single characters and we run the experiment, we'll end up with an approximately 3 to 1 ratio for that exact same reason. Each of these is an allele residing on a different parental chromosome. And when we cross, we have first of all an F1 generation, then we go to the F2 generation, they resegregate in such a way that the recessive reappears. The recessive reappears here in the homozygous form. Okay, let's see. I think this is not, you're not going to be able to hear. I think that's a problem, but nonetheless, sorry, let's try and do it. I don't know why these things really. Oh, what did I do? Sorry. Sorry. Okay. Okay. So here's again, here's a summary of some of the terminology for you. If an organism has two identical alleles for a particular gene, for a particular character, that's called the homozygote. And it's a heterozygote if it has two differing. In Mendel's experiments, they were clearly on or off, either purple or white, either round or wrinkled, etc. And in his experiments, they inherit these either in the homozygous condition, or we inherit in the heterozygous condition. The heterozygote has differing, it has a dominant and a recessive inherited together. That would be a heterozygote. So again, I'll just point out to you, going back, we said that Mendel always chose true breeding plants. So you had purple, true breeding. What that meant, now we can understand that in fact the plants that he started off with were homozygous. So the parental plants, purple plants, were homozygous purple, homozygous dominant. The other plants, the contrasting plant, the white, is homozygous recessive, the recessive gene on both maternal and paternal chromosome. The reason we say that the heterozygotes are not true breeding is because when you breed the heterozygotes together, you get this 3-to-1 ratio, the recessive reappearing as white in this case, and so again against 3 of the purple. So and this also bears emphasizing is this distinction between the genotype, which is the true genetic makeup of the plant, and its phenotype, what we observe. That purple phenotype we've seen can be produced by 2 genotypes. It can be produced by the homozygous purple, homozygous dominant, or by a heterozygote. So it's either big P or big P little p. They produce the same phenotype, but the genotypes are quite different. Okay, so here's our 3-to-1 ratio, explaining this exactly. The phenotype, these are all purple. So they're all the same phenotype. There are 3, they appear in the 3-to-1 ratio. We would have 3 purples to every 1 white. And that purple condition can be produced here by the homozygous, big P, or it can be produced by the heterozygous condition, big P, little p, big P, little p. They're in our breeding experiments. There are 2 of these, 1 of these, and then 1 homozygous. And there you see that there's actually another little ratio built in here, which might be worth remembering. And that is, there's a ratio of homozygotes to heterozygotes, 50-50. 1/2 of them are homozygous, 1/2 of them are heterozygous. And these heterozygotes look exactly the same as these homozygotes. The only one which looks different is this homozygote here, which is the homozygous recessive, which gives you the white flower. So what we have actually is a 1-to-2-to-1 ratio, but we observe these as being identical in the phenotype. So that is observed as a 3-to-1 ratio. So the phenotype and the genotype can be quite different to one another. So this presents us with a little bit of a problem, and that is that sometimes we'd be looking at a plant and we don't know what its genotype is. We can't tell what the genotype is just by looking at a plant. Give you a purple flowering plant and it could either be a homozygote or it could be a heterozygote. Whenever you're presented with that problem, there is a simple solution, and that is to do what is called a test cross. And that is to cross that plant, the suspect heterozygote, to cross that plant with a homozygous recessive individual. And any offspring if any offspring show the recessive phenotype, the parent plant must be heterozygous. So here's how it works. So here we've got, here's the purple plant. It's what can it be? It could either be big P, or little p, either homozygous or heterozygous. We don't know. And in order to tell, we're going to cross it with a true breeding recessive. So we know that this is a homozygous recessive, little p. Now let's run through the different possibilities. Well, if this purple plant is homozygous, the homozygous dominant, let's say we're going to bring cross pollen from the white flower, right? So here's the possibility for the purple flower. We're saying it could be homozygous, big P. We know that the only pollen which this plant can produce is little p, because it's homozygous recessive. So here's the pollen, here are the eggs, big P. Here's the pollen, little p. If all of them turn out to be heterozygous, big P, little p, big P, little p, big P, little p, big P, little p. What we would observe if we did this experiment and we did that mating and we grew them out, we would immediately see there's no white at all in this generation. There is absolutely no white. All the offspring are purple. And that immediately tells us this had to be homozygous dominant. It had to be big P. That's the only way to account for the fact that all of the offspring are the purple phenotype. What about if this plant is in fact a heterozygote? Well, if it's a heterozygote, here's the possible eggs, big P, little p. We know, because we chose it, that this plant is homozygous recessive, little p. So here's the only possibility for the pollen, little p. What is the possibilities for the offspring? Big P, little p, big P, little p, little p, little p, little p, little p. If this is a heterozygote, big P, little p, the white flowers will appear, and they will appear in a 50-50 ratio. We'll go to when they start flowering, we'll go out to our field and we'll see that half of them are purple and half of them are white. The parent was a heterozygote. If, on the other hand, all of the offspring are purple, the parent was a homozygous dominant. So that is a typical test cross when we don't know the genotype, even though we can observe the phenotype. So this law of segregation was derived by following a single character in the breeding experiments. And when we look at that, when we're taking purple and white wrinkles, smooth, green, yellow, and we're crossing them, the F1 hybrid is called a monohybrid. That we're looking specifically at one character. And in that F1 generation, they were heterozygous. They were all heterozygous for one particular character. We call that we say they are monohybrids. And then the F2 is a monohybrid cross. But it's really important to realize that the plants can have many characters, many different characters. And how do we account for the interaction of different alleles with one another when they apply to completely different characters? If I have a purple flowering plant, which is a dwarf, and I mate that with a white flowered plant that is tall. How do these four now interact with one another? And was Mendel's second law of inheritance, which he derived by crossing plants with these two characters, and those he referred to as dihybrids. So when we take plants with two characters, two corresponding characters, mate them, F1, we end up with a dihybrid. We then allow first cell fertilization. That is a dihybrid cross, a cross between F1 dihybrids. And we now are going to watch and see, are characters handed on together, or are they handed on separately from one another? And I think I'm going to stop here because I'd rather discuss this in our next class. So for the whole of this portion of the genetics, you need to read carefully in your book, but also please refer back to what we learned on mitosis and meiosis, because if you can link this to the processes of meiosis, you will understand the mechanisms of this very much better. Okay, and then I'll see you again on Monday.